All right. Hello, everyone. Today we're going to be doing the gene expression lab. All right. So I'm going to go back. This is lab 8, gene expression. I'm going to explain the experiment in detail, show you guys results, and you guys will fill out the test sheet using that. All right. So for today, we're going to be talking about plasmids and bacteria. So what is a plasmid? It's basically a circular piece of DNA that exists separately from the chromosomes of bacteria. All right. So the thing about this is that sometimes or well, the majority of the times it's not really essential to bacterial survival. There are some instances in which, for example, the one that we're going to look at today, a plasmid can actually help a bacteria survive. But in most cases, they're not essential to bacterial survival. So let's talk central dogma. We kind of went over this last week. It's the process in which we make proteins using our DNA. So it goes DNA to mRNA, that's transcription. And then from mRNA to a protein, it's translation. So basically today, you're going to see how a bacteria takes up a foreign piece of DNA, expresses it, transcribes it, and translates it into a protein that allows it to be antibacterial resistant. Okay. So this is a form of biotechnology. Essentially, you modify your genome in order to produce a specific thing or a specific process. All right. So there's a few examples of this. In healthcare, for example, we can use it to manufacture hormones like insulin. And then basically all of these other examples. It's a super important tool that we have to do basically all of these things. Right. And these are all of the examples that we have here. This is all created or was created using biotechnology. Okay. So today we're going to be talking about this plasmid PUC18. We're going to be basically introducing it to E. coli, and it encodes for antibiotic resistance. In this case, for E. coli, it specifically makes it resistant to ampicillin. Okay. So essentially, in order to make sure that the bacteria E. coli is able to acquire that PUC18 plasmid, there's a few things we have to do. All right. So the first thing, we have to make sure that the E. coli cells are competent. To make a cell competent, you basically just neutralize the charge of the cell membrane in order to allow it to more easily take up a plasmid. So in this case, the bacteria that we used in this lab is already competent. They were giving a calcium chloride bath in order to neutralize the charge of the cell membrane and allow it once again to uptake that plasmid a lot easier. Okay, so make cells competent. Again, this was already done. The second thing that we do is a heat shock. For a heat shock, in this case, what we did is we put the agar plates in the incubator for 30 minutes to let them get super toasty. And then we have our plasmid and our bacteria on an ice bath. That cold ice into that hot agar plate causes a heat shock, which opens up the pores of the cell membrane and once again allows the E. coli to uptake that PUC18. Okay. So the making the cells competent, that was already done for us. All we did in this lab was the heat shock. So we actually went ahead and put the agar plates, made them hot, and then got our plasmid and our bacteria from an ice bath. So how do we assess transformation? In this case, we're going to have two controls and two treatments, and they're going to look a little bit like this. So these are the 4 agar plates that we prepared. The treatments are basically those that are just grown on LB plates. LB is Luria broth. It's just food for the E. coli to grow. All right. So if the plates had no LB, there would be no growth at all. All right. So we had 2 plates that were LB and then 2 plates that had that LB. And on top of that, were also infused with ampicillin. What we normally see is a plate with ampicillin will kill off E. coli completely because, again, E. coli is killed off by ampicillin. However, when we give E. coli that PUC18 plasmid, it should be able to fight against that ampicillin that's infused into the plate. All right, so what we're expecting to see is this plate, LB and P. NP stands for no plasmid. So this plate is just getting regular E. coli. You should see that the E. coli grows all over the plate, no restriction. It's just a plate that has food. Given an E. coli, it grows. Okay. In this case, LB and plasmid, this plate once again has that broth, that food, and you introduce that E. coli with the plasmid already incorporated into it. What should happen is the same thing that happened in the other LB plate, just uncontrolled growth. Remember that we mentioned earlier, plasmids aren't essential for bacterial survival. So in this case, since E. coli is not fighting against anything, the plate just has LB. It's just regular E. coli with that added plasmid. It's not giving it any sort of advantage. For this plate here, we have LB, and again, this plate is infused with ampicillin. However, you're dispensing regular E. coli with no plasmid into here. What should happen in this case is since that E. coli has no way to fight against the ampicillin, there's no growth. The bacteria is completely killed off by the ampicillin that's infused into the plate, and you see no growth happen. All right. For this bottom one here, you have LB amp, and you have that plasmid incorporated into E. coli. Like we mentioned earlier, this PUC18 plasmid is known to give E. coli that ampicillin resistant. So in this case, we should see some growth. The issue is not all of the cells are going to be able to uptake that PUC18 plasmid. So it's not going to be this full free-floating growth that we had for the 2LB plates. Instead, they're going to grow in colonies, where the bacteria that was able to take up the plasmid survives, and then the bacteria that weren't able to take up the plasmid are killed off, right? So for this one, we should have colony growth. Colony growth, and then these two that were able to grow unrestricted, we call that lawn growth. And those are the results that we should see. I'm going to show you guys pictures of what it actually looks like on the agar plates. But let me clear this so we can keep going. All right, this is the setup that we did. Again, incubated the plates at 37 degrees for 30 minutes. That's to make them hot in order to obtain that heat shock. Okay, this was the setup that we actually did. And then this is your lab log. All right, so like we said, we predicted that the LBNP is going to have lawn growth. This one, the LB ampicillin plus no plasmid, had no growth because, again, that ampicillin killed off the E. coli. The LB plate that had the ampicillin, that had the plasmid in it, was just once again lawn growth. And then the plate that had the ampicillin and the plasmid, we saw colony growth. All right, that was what we predicted. The results should match up. And then for the description, you can just talk about what kind of growth it is. So once again, lawn growth is that uncontrolled growth. No growth, the plate was just empty. Lawn growth is uncontrolled growth with E. coli covering the entire plate. And then I'll show you guys what colony growth looks like on an actual plate. It just shows up as a bunch of dots. Okay, so you guys should be able to fill this in. I'm going to show you guys some pictures that you can include on your lab log. But if I'm not mistaken, the lab log says that you have to draw the actual plates, so you should just be able to do that. All right, so these are pictures of the actual plates. I'm going to zoom in here. This is my LB plate that had the actual plasmid into it. So if you zoom in, you can see lawn growth. You're going to see little like scratches in it. That's just because whoever spread this plate kind of jumped over a little bit. But as you can see, the bacterial growth covers the entire plate. So in this case, again, this is just a Luria broth plate with E. coli that had the plasmid. Again, the plasmid provides basically no advantage in this case because it's just growing on a regular LB plate. So you just see uncontrolled lawn growth. All right, this next plate is another Luria broth plate. This one had no plasmid. So this is just regular E. coli. And again, you can see that complete lawn growth. This was spread a lot more evenly. And again, the E. coli covers the entire plate because there's nothing to fight off. It's just LB. There's no ampicillin in this plate. You see these little white colonies here. That is contamination. That's not E. coli growth. This third plate is my ampicillin plate that had no plasmid. All right. So in this case, this is just regular E. coli put onto an ampicillin plate. As you can see, there's no growth because the ampicillin completely killed off the E. coli. The E. coli didn't have that PUC18 plasmid to help it fight back against the ampicillin. So you just see a clean plate with no growth. Okay. This last plate is my ampicillin plate that had that plasmid. All right. So in this case, the bacteria that was spread onto this plate was E. coli that had uptaken that PUC18 plasmid. And as you can see, it grew in colonies. The bacteria that were actually able to uptake that PUC18 and incorporate it into its genome was able to fight back against the ampicillin and grow. But again, it's not long growth because some of those cells weren't able to take up the PUC18 plasmid. All right. And that's basically the entirety of this lab. You can go ahead and start working on your task sheets now. You have all the material to fill it out. And yeah,